Let's start our lesson today about lipids. Lipids are the second type of biological macromolecules forming the chemical structure of living organism's body. We talk about the carbohydrates. Now we are going to see the lipids. What is lipids? Lipids are biological macromolecules, polymers also, which is formed of smaller molecules or units, monomers. These monomers called fatty acid. So the carbohydrates formed of monomers called monosaccharides, and the lipids formed of fatty acids. The lipids consist of carbon, hydrogen, and oxygen atoms, but not by the ratio 1 to 2 to 1 like the carbohydrates. They have different ratio. Their molecular structure is formed of 3 fatty acids and 1 glycerol molecule. Glycerol is a type of alcohol formed of 3 hydroxyl group. So we have 3 fatty acids in the structure of any lipid. What are the types of lipids, or what is the classification of lipids? The lipids is formed of simple lipids, complex lipids, and derivative lipids. What is the difference between them? That's what we are going to talk about. But first we are going to talk about simple lipids. How they are classified, or how the simple lipids are formed. They are formed of reaction of fatty acid with alcohol. They are divided according to saturation degree of fatty acid and type of alcohol. So when the saturation or the type of alcohol change, the type of simple lipid change. What is the meaning of word saturation? Look at these two molecules and look at the bonds between the carbon atoms. The chain of carbon atoms of fatty acid here and chain here. Here all the bonds are single bonds, and here we have one double bond. So the number of hydrogen molecules here more, and the number of hydrogen molecules here less. So when we have double bond between the carbon, it is called unsaturated, and it becomes saturated when it is all the carbon have the maximum number of a hydrogen linked to it, and we have only single bonds between them. So this is called saturated fatty acid, and here this is called unsaturated fatty acid. The first type of simple lipids, we are going to talk about oils. Oils at ordinary or at room temperature, they are liquid fats. They are formed by reaction between unsaturated fatty acid with glycerol. So they are unsaturated fatty acids. Example for the oil are the oil that cover the feather of water birds. The water birds, they have a layer of oil covering their body, covering the feather of their body. This oil prevents their body from water to enter inside it. It prevents water penetration to their body, so they can move more easily. When the water enters their body, it hinds their movement. So it is layer for protection. So the first type of simple, they are liquid, which are oil. They are formed of unsaturated fatty acid. Now the second one, which is fats. Fats are solid substance at ordinary temperature or at the room temperature. They are formed of reaction between saturated fatty acid, and when the molecule becomes saturated by hydrogen molecules, they become solid. So they are called fats. And the example for the fats, like the fats stored under the skin of some animals, especially the polar animals. This layer of fat acts as thermal insulator. They keep their body temperature. They can give them the ability so they can live in severe cold regions. The third type of simple lipids is wax. Wax is also solid, but it is formed by a reaction between high molecular weight fatty acid with monohydroalcohol. So they are formed between high molecular weight fatty acid and monohydric alcohol, and this alcohol contain only one group of OH, one hydroxyl group, not like the glycerols contain 3 hydroxyl group. We can find the wax over the leaves of the plants, especially desert one, because they act as a layer keeping the water inside this tissue and reducing the transpiration, reducing the movement of water away. So they keep them. They reduce the water loss inside, especially the desert plant, this wax layer. The next type is complex lipids. Complex lipids are formed of carbon, hydrogen, oxygen, but we have additional molecules here. Phosphorus and nitrogen may involve in their structure. So we can add sulfur and phosphorus or nitrogen inside their structure. Example for the complex lipid, we are going to have phospholipid. So from the word phospho, we have here phosphorus. They are present in the cell membrane of some animals and plant cells. Their molecular structure of fat molecule, but they replace the third fatty acid with phosphate group and choline group. We have 2 groups replacing the third fatty acid. One is called choline group and the other is called phosphate group. It consists of 2 fatty acid and glycerol molecule and phosphate and choline group. So the complex lipid like the simple lipid, but the third fatty acid molecule is replaced by phosphate and choline group. So we have only how many fatty acids inside the complex lipid phospholipid? We have 2 fatty acids and glycerol molecule and phosphate and choline group. The phospholipids enter mainly in the formation of the cell membrane structure. They enter mainly in the formation of cell membrane. Now we have derivative lipids. The derivative lipids, they are derived from both simple and complex lipid by hydrolysis reaction. What is the meaning of hydrolysis reaction? This reaction occurs by adding water. When we add water, we have hydrolysis reaction. We have example for the derivative lipids: cholesterol, hormones, steroids. So we have 3 types of lipids. Simple lipids are divided to oil, fat, and wax, and we have complex lipids like phospholipids, and we have derivative lipids like cholesterol and some hormones and steroids. The lipids have a property: they are soluble in non-polar solvent and insoluble in water. Any lipid cannot dissolve in polar solvent like water, but they can dissolve in non-polar non-polar solvent like carbon tetrachloride or benzene. Non-polar solvent like benzene and carbon tetrachloride. Now we are going to see the importance of lipids. Why lipids are important in the structure of living organism? The first importance is obtaining energy. Lipids, especially fats, are an important source for obtaining energy, but doesn't begin to get energy from fats stored unless there is no carbohydrates. When we have carbohydrates inside our body, we are using carbohydrates as source of energy. But when the carbohydrates run out of our body, the body start using the lipids as a source of energy. The amount of energy obtained from lipids is more than obtained from carbohydrates if they are the same amounts. If we have one mole of lipid and 1 mole of carbohydrates, we get more energy from lipids, but we get faster energy from carbohydrates. The second importance, building cells. Lipids represent about 5% of the organic materials that are involved in the composition of living cells. Lipids form, especially the phospholipids, the structure of cell membrane or plasma membrane. The third importance, it works as thermal insulator. Lipids or fats especially are insulating layer. They are found inside the skin of the human or skin of animals, especially the poorer animals. We have a thick layer of fats under their fur to keep their body temperature in cold, severe regions, so they can overcome the low temperature and protect them from it. The fourth importance is it works as protective layer, like the wax. The wax covers the surface of some plants, especially desert ones, so reduce the water loss by transpiration and protect them from dryness. And the last one is it works as hormones. Lipids can work as hormones and as steroids. So these are the importance of lipids. Now we are going to see how to detect the presence of lipids in the substance. We can detect the presence of lipids by a special reagent which is called sodium 4. Sodium 4 is a stain that is used for detecting fats in different food. The color of sodium 4 is changing to red in the presence of lipids and fat. The sodium 4 becomes stained red in case of the presence, so it change its color red. Sodium 4 is used to detect fat in different food like oil, milk, and peanuts and butter because its stain is soluble in fats, where it turns into red and color presence of fats and lipids. So this is the second type of biological macromolecules, which is lipid. We can see here I have some examples for different types of lipid, and there are different sources of fatty acid, and we said that the saturated with no double bond, and we have unsaturated and they have double bond, maybe one or more double bond, maybe monosaturated or polysaturated. And we have here example of sources of saturated like red meat, coconut oil, and dairy products. And we have here monosaturated or polysaturated like olive oil, canola, avocado, and they are different, and nuts and corn oil, sunflower. These are different types of food containing saturated and unsaturated fatty acid type. Thank you and have a nice time.